You are listening audio paper series with Yasir, and today's guest is Dr. Thomson from Quadram Institute, Norwich, UK, to discuss one of his publication, Creation of Golden Gate Constructs for Gene Doctoring. It is published in BMC Biotechnology. Welcome, Nick.
Hello.
Hello, Nick. Would you like to tell us the aim of this project?
Well, the big idea was that basically to make it easier to manipulate the genes on the chromosome of bacteria, really focusing on E. coli, but not necessarily limited to that species. And up until now, gene knockouts particularly have been very widely used for testing the functions of genes. But the process of making a gene knockout is usually very inefficient because it involves transforming the cells with a linear DNA molecule. A while ago, there was a technique that was developed called gene doctoring, which uses only circular plasmids for the transformation stage, and this makes getting the knockout construct into the cells a lot more efficient. But the process of actually making those plasmids was quite inefficient also. It involved quite a lot of cloning steps. And so I wanted to use a more streamlined method of generating the plasmids in order to do the gene doctoring, so that both stages of creating knockouts or for putting new genes into the chromosome would be more efficient. And so for that, I used a technique called Golden Gate cloning, which allows you to join a pretty much unlimited number of parts in a single reaction.
Nice. So your method is based on gene doctoring. What modification have you done to achieve this version?
So for gene doctoring to work, you need two plasmids. There's one that introduces the knockout cassette, and then there's another helper plasmid that expresses the lambda recombinase system. And what I focused on was the first plasmid for introducing the knockout cassette. And basically, I made three major changes to that plasmid. The first was very often Golden Gate libraries are maintained in plasmids that have beta-lactamase resistance, ampicillin resistance. But the existing Golden Gate plasmid also had ampicillin resistance, and so I switched the resistance cassette marker on that to kanamycin, which makes it compatible with existing libraries. I also introduced a LAC-Z alpha fragment into the cloning region, which means that successful clones can then be selected by blue-white screening. And I also removed a BSA1 restriction site from the existing Golden Gate, the existing gene doctoring plasmid, which made it more compatible with Golden Gate cloning.
So in summary, you have changed the selection cassette, introduced LAC-Z, and removed BSA1 restriction site?
Yes.
Nice.
And also introduced two new BSA1 restriction sites in the correct place to allow for the insertion of the new knockout cassette.
Oh, okay. So these BSA1 restriction sites are particular to Golden Gate construct.
So BSA1 is a specific type of restriction endonuclease called a type 2S enzyme. And unlike the more commonly used enzymes, it doesn't cut within the recognition sequence, but it cuts a few base pairs downstream of the sequence. And therefore, it doesn't have a defined overhang that it leaves in the DNA. You can edit the DNA in any way you like to tailor the overhangs to fit with your parts, and that's what allows you to direct the assembly of the parts in a particular order.
After these modifications in gene doctoring, what are the main application of this construct?
The main applications will be either for knocking out or inserting genes on the chromosome. So in the first case, knocking out genes would mainly be used for testing the function of a gene that you might have identified through some previous experiments. And for inserting a gene would either be if you want to put a marker into the cells, or possibly for some sort of strain engineering type applications, where usually inserting genes onto the chromosome is a lot more stable than maintaining them on plasmids.
You mean it can be used for gene doctoring purposes, but with ease of cloning?
Exactly, yeah. It basically makes the entire process a lot more streamlined and efficient, and it should be a lot more applicable to high throughput applications.
Thank you, Nick, for sharing this amazing update.